At first glance, this may just look like a moving shape under the microscope, but this is actually a protozoa.
Protozoa are unicellular eukaryotic microorganisms that are widely distributed in the environment and play an important role in nutrient cycling, microbial regulation, and ecosystem balance. Through this project, we aim to isolate and observe protozoa, focusing mainly on their morphology, movement, and different stage under laboratory conditions. So hello everyone, my name is HANIS and today me and my friends will explaining to you about the sampling of protozoa.
Hi, my name is ZULFA Mustafa and my matrix number is S72467. So for methodology, I will explain to you about the brief explanation. So for the first one is for the study design. For this study, a laboratory-based experimental design was used. The aim was to isolate, cultivate and observe protozoa from different environmental sources. The second one, which is sample sources, protozoa samples were collected from stagnant water, laboratory water and soil. These environments were chosen because protozoa usually live in moist area with high microbial activity. Next is a sample collection. All samples were collected using aseptic techniques to prevent contamination. Water samples were collected using the containers. Soil samples were taken from the upper soil layer using sterile cotton. Next one is the sample preparation. Water samples were directly inoculated onto the non-nutrient agar plates. Soil samples were mixed with sterile distilled water and left to settle. And the clear supernatant was then used for inoculation. Next is cultivation of protozoa. The inoculated plates were incubated at room temperature. Plates were observed daily for signs of protozoan growth. And solid agar was used to slow down protozoan movement. This allowed clearer observation of their structures and movement. Changes in growth pattern and life stages are also monitored. The next one is microscopic observation. Protozoa were observed using a light microscope and wet mount slides were prepared from agar sample. Cell shape, size and movement were carefully examined and the internal structures such as vacuoles and nuclei were observed. The last one is data recording. All observations were recorded systematically in tables and microscopic images were taken where possible. Results from different samples were compared and using standard procedure ensured the results were reliable and reproducible.
In both stagnant water sample, we identify organisms from the genus Amoeba. In their active stage, they appear asymmetrical and irregular. We clearly observe amoeboid movement, where the cell use cytoplasmic streaming to push out the pseudopodia or false feet at the leading edge. While the uroid trails behind, you can see the granular endoplasm in our micrograph, which contain the organelles. We also discover this organism in their dormant and cyst stage. Unlike the active cell, the cysts are round and immobile with no pseudopodia. In our lab samples, we identify acanthamoeba cysts. They have double layer walls, a wrinkle fibrous ectocyst on the outside and endocyst on the inside. This structure protects the condensed cytoplasm inside. To study stagnant water, we choose to grow our samples on solid agar rather than in liquid. This is because amoeba moves slowly. The agar acts as a stable stage, allowing us to clearly see the false feet pseudopodia and identify their front and back end without them drifting out the view.
In this study, protozoan samples were collected from two types of stagnant water, rainwater in a drain and water from a pond. Both environments support protozoan due to stable condition, available nutrient and minimal disturbance. Under the microscope, amoebas were observed in both samples with similar morphology. They are unicellular, colorless, jelly-like organisms ranging from 250 to 750 micrometers in size. The external features include pseudopodia for movement and feeding, a plasma lemma for cell integrity, and a uroid for waste removal and adhesion. Internally, amoebas have nucleus, contractive vacuole for water regulation, food vacuoles, and water globules, all supporting movement, feeding, and survival. Overall, despite slight differences in their habitat, the amoeba show similar structure and adaptability, reflecting how they thrive in stagnant freshwater environment.
Now, I will briefly explain the life cycle of protozoa. Protozoa have two main stages in their life cycle, the trophozoid and the cyst. The trophozoid is the active feeding stage inside the host. When conditions become unfavorable, it forms a cyst to survive. The cyst is dormant and helps in transmission. Once inside a new host, the cyst releases the trophozoid and the life cycle will continue.
Next basis for the discussion, protozoa are scientifically important because they are single-cell organisms with a very simple structure. This makes them useful for studying basic cell processes. Scientists can easily observe movement, feeding, and internal cells activity using a light microscope. In this study, amoeboid protozoa were observed, which are commonly used to study cell movement and phagocytosis. One important feature of protozoa is the ability to form cysts. Encystment allows protozoa to survive harsh conditions like lack of nutrient or environmental stress. During the study, cyst forms were observed in several samples. This showed that protozoa can adapt and survive until conditions become favorable again. Ecologically, protozoa play an important role in the environment. They feed on bacteria and algae, which helps control microbial population. This feeding activity also helps recycle nutrients such as nitrogen and phosphorus. As a result, protozoa help maintain balance and stability in the ecosystem. Protozoa are also useful as bioindicator of environmental quality. Changes in the presence of life stages can indicate pollution or environmental stress. In this study, the presence of encysted protozoa suggests that some environments were under stress. This shows how protozoa can reflect changes in environmental conditions. Protozoa are also important for public health. Some protozoa can carry harmful bacteria inside their cells. This allows bacteria to survive in water system even after treatment. The presence of protozoa in water samples highlight the importance of regular monitoring. In conclusion, protozoa are important scientifically and ecologically. They help scientists study basic cell processes, regulate microbial population, and indicate environmental health. Their presence in different samples show their adaptability and significance in environmental studies. Thank you.
So in conclusion, this mini project successfully showed that free-living protozoa, particularly amoeba, are present in common environmental water sources. The observation of both active and cyst stages highlight their adaptability and survival strategy under environmental stress. Overall, this study emphasized the ecological importance of protozoa and their potential use as bioindicator for environmental monitoring and also water quality assessment. That's all from us. Thank you.